Welcome to the Radiation Research podcast of April 2012. Today we are going to talk with Rao Papineni and Mansoor Ahmed, and the title of their paper is A Realistic Utilization of Nanotechnology in Molecular Imaging and Targeted Radiotherapy of Solid Tumors. It's a review on basically the utilization of nanotechnology and radiotherapy. So, Rao and Mansoor, welcome.
Thank you. Hi, Sylvain.
Maybe you want to introduce yourself first, since you're in two different institutes.
I am Mansoor Ahmed. I'm currently with the Radiation Research Program at the National Cancer Institute in National Institutes of Health.
I am Rao Papineni, research scientist, senior principal investigator at Carestream Molecular Imaging, Carestream Health, USA.
Thank you. Well, welcome to both of you for this discussion on your work. So, in this review, you discuss the usage of nanotechnology for both imaging and radiotherapy. So, my first question for you is, what was the motivation behind writing this review?
Okay. So let me answer that. This is Mansoor, actually. So before I get into this, I just wanted to do a disclaimer. Since I moved to NCI, I have to disclose that whatever I say here, it is solely my own knowledge-based opinion. It's not the opinion of NCI or NIH. So having said that, this particular article, actually, I was a member of the Bridge Group in Radiation Research Society where Mark Dewhurst was the moderator. So there was a plan within the Bridge Group to bring out a special issue on the imaging in radiotherapy, particularly in preclinical as well as in mostly giving thrust to the preclinical model where there's any new advances that is happening there. So during the course of the discussion, I sort of working in the area of targeted imaging of solid tumors, particularly in prostate cancer, where we wanted to use nano-based approaches. So that's where I sort of indicated to Mark, and Mark came out with the idea, why don't you write this review article? So I sort of brought in, as I was collaborating with Dr. Papineni at Kodak using his nanoparticle. So we wanted to group this together and write this up. And again, there are a lot of various imaging modalities currently available, but the fluorescence-based, where there are a lot of things happening using nanoparticles, and that's where I brought in Rao, and we wrote this article together.
Rao, do you want to add anything?
Yes, as Dr. Mansoor Ahmed was mentioning, ongoing collaboration with the University of Miami is going on with some of our expertise. That is Carestream. We're primarily the first to bring in a multimodal technologies in preclinical. So there's a perfect match here to discuss about components like nanotechnology and molecular imaging, because they form a basis of what you call diagnostics. So that's, again, a good motivation, and it's a good collaborative review article here.
Okay. Let's start with the diagnostic side, the imaging part. So you illustrate how nanoparticles can be used to enhance imaging.
Yes. This is a very interesting question, Sylvain, especially in a rapidly growing field. What one can bring in with this nanotechnology-based component in imaging is it provides, in the case of preclinical at least, a very sensitive definition of the target. You can actually direct what to image by providing a targeting agent to some of these nanoparticles. It could be a polymeric nanoparticle or a metal-based gold nanoparticles, etc. So one advantage over this is, apart from diagnostic purpose, it has become, and it is becoming, a component in a multifunctionality, which we can discuss in the next few questions. And if Mansoor wants to add anything to it, please do so.
Well, I think there are current technologies available in terms of diagnostic imaging, but the question is, there's always an issue to look into the clear tumor margins. Can we clearly demarcate? So most of the time when you look at physicians or the radiologist or radiation oncologist marking the tumor area, it is only arbitrary, but you cannot really precisely define those margins. So that's one of the reasons I think the nanotechnology played an important role. And again, with the fluorescence-based imaging, sometimes the image taken, particularly, it doesn't work with the deep-seated tumors, even if it is a superficial. But at the same time, you cannot get a sharp, focused image. So there are a lot of things happening in the field, and that's where I think Rao's company has done more kind of diagnostic-based imaging using nanoparticle in a fluorescence setting. So I think this merging of these areas of the current modalities, such as the MRI-based or the CT-based or any other metabolic or molecular imaging based that is currently used in diagnostic imaging, can be coupled with the nano-based approaches and coupled again with the fluorescence-based approaches. I think that will synergize more exact defining point between the tumor and the normal tissue. So that's the whole idea where we had to move into to write this review article so that we can trigger thoughts among the readers who like to read this article.
I'm going to try to summarize very briefly the idea here, which is to use nanoparticles and have them binding specifically to tumor cells. You described as two possibilities. You can do it in a passive manner, or you can actually use active nanoparticle that recognizes very specific property of the tumor cells. So how specific are they compared to other type of approaches like chemical approaches?
I will tell about this at the same time. I'll ask Rao to feed in on to this. So basically, we cannot use just the nanoparticle alone. We have to modify, do some chemistry in order to play around. So here we are doing two important things. One, nanoparticle is used mostly as a vector to carry two important things. One is a targeted agent that will specifically identify that particular aberrant tissue. Like, for example, if you take prostate cancer, in prostate cancer there are a lot of surface antigen expression that is present, which is one of them is like prostate-specific membrane antigen. So there are antibodies that can basically target this antigen. So what we did was that in this particular instance, we can basically load this nanoparticle with an antibody against the prostate-specific membrane antigen. And when you use this nanoparticle or inject them in the tail vein, even though it goes initially to the liver, but it slowly starts to accumulate only in the prostate tumor region. Now, in fact, we use a xenograft orthotopic model where we injected the human LN cap cells that expresses high levels of this antigen PSMA, and basically when you inject this nanoparticle that contains this, the PSMA antibody, it goes and sort of accumulates over the time into the tumor area. So the advantage, since it gets targeted so specifically into that area, so we loaded the nanoparticle with a therapeutic agent, like an siRNA against MDM2 or any other thing. You can put a plasmid or a protein into that so that you can easily target and burst this nanoparticle into the tumor area. So it's more of a very highly targeted approach that can be done in a mouse model. Now, I leave it to Rao to add more onto this from here.
Yeah, the difference when it comes to a nanoparticle compared to a traditional approach, number one is the lingering time or the residence time of a nanoparticle is much higher in circulation. That allows more opportunity for this targeting agent or a passive agent to get to the site of interest. When you have the same, like what Mansoor was mentioning, when we have the anti-PSMA recognizing nanoparticle, this has the capacity to, in the case of a mouse, for example, it has the opportunity of visiting the site every 8 minutes, while an antibody by itself, it leads to potential clearance, increased clearance rates, and non-specific binding at other cellular regions. So that, in a way, is an interesting aspect, which is going to be beneficial not only in the preclinical, but can be translatable from into a diseased scenario in human health.
I suspect since those agents are able to stay longer, is there any toxicity involved with nanoparticles?
Well, the review is concentrating on almost each and every corner of the nanotechnology, especially biomedical applications. Again, these nanoparticles need to be very well characterized, whether it is a polymeric nanoparticle, which is pegylated, or a PLGA, which is probably one of the best nanomaterial, or a gold nanoparticles or quantum dots, which is now a modified version. So again, the toxicity, cellular toxicity, tissue toxicity, and animal toxicity will be unique, and it will be different from a particle to particle. And on top of it, when it comes to multifunctionality, or adding a targeting agent, or adding a drug encapsulation inside, or putting a new reporter system outside on the nanoparticle, decorating these components will actually add at every facet additional question raised, whether it is toxic or whether it has any deleterious effect. So all these things have to be considered as one takes these steps.
The nanoparticle where Rao's group is developing, where I have used it in the lab, had a very minimal, raise in the liver enzymes, but otherwise I didn't see any big changes. So I think Rao's group have done extensive normal tissue analysis in that labs before it comes out. So I'm sure, it's not like quantum dots sometimes, has more higher toxicity. So that's the whole idea, to make nanoparticles that are less toxic.
Yes, there are several biocompatible nanoparticles coming into the picture. They are very useful and less toxic, especially biomaterials, which is already existing within the body, has been utilized to develop nanoparticles. Like simply, for example, albumin has been taken into picture, or polylactic materials. So there are very good amount of biomaterials available.
I see. And in terms of clearing those nanoparticles, once you have them inside your system, do you have any idea how long it takes for them to be cleared entirely from your body?
Again, to answer the question, the clearance rates depends on the biomaterials. The current the new trend in nanotechnology is focusing on the idea about until what time you want to have these nanoparticles present inside and when to degrade. Again, you can have all these designed something like an intrinsic destructive elements can be put in within these nanoparticles, which can allow clearance at a much faster rate if you need it, or on a timed manner.
That's very interesting. It is a very flexible system. So to move forward with this discussion, the next idea. So to summarize so far, you can use nanoparticle to enhance the definition of the tumor boundaries, which would then help you have a better idea where the tumor is, and you can get better treatment planning with radiotherapy. Now, the next step is you could also use those nanoparticles to enhance the radiotherapy. Is that correct?
Absolutely.
How does that work?
So one good example is the gold nanoparticle. This actually increases the radio sensitization with a photodielectric effect. So the killing capacity of these nanoparticles is currently studied by multiple groups. And I'm sure Mansoor can add more on a clinical perspective.
Yeah, no, basically the whole idea is, I think we can, I'm not sure, maybe Rao can answer this, but the way where we were trying to use is more the gene products, or manipulating at the DNA level, or manipulating at the RNA level, or putting a protein product that can radiosensitize the effect at the tumor level. So one thing is the nanoparticle that has a specific targeted agent basically goes and binds to that region, and this nanoparticle itself is more of a fluorescent-based nanoparticle that can be excited. And most of the best way to use a nanoparticle that can be excited is around near infrared range, and this is more important so that you eliminate all the background. You are just seeing the tumor glow there with the targeted agent, but at the same time, you have already loaded in certain protein modifying agent, or the RNA silencing agent, or DNA switching off agent, something like that. So basically, the whole idea is, I was using in the lab, it's more of either a siRNA or a protein or a plasmid that can be, loaded up into this nanoparticle. But I think Rao can even talk more about, can we put some, agents into it, like a chemical agents into it.
Before you go there, I had a question about this. So typically to do siRNA or any kind of work like this on human cells, epithelial cells tend to be quite resistant in being transfected. And in the lab, we use viruses, for instance, to get a better infection rate. So has it been shown that nanoparticles are actually better carriers than viruses, for instance?
Yes. Like we all know, delivering gene has been done for a long period of time using even liposomes, etc. Nowadays, I think we have certain kind of like chaperones which allow the nanoparticles to be facilitated inside the cellular components and then released either siRNA or shRNA, whatever one has designed. in a specific manner. So I think the what you call as a transfecting rate is probably enhanced with the current models like multifunctional nanoparticles.
I see, but one of the issue with classic plasmid is that as they get inside the nucleus, they very quickly get removed because they're not really well incorporated in the genome. In contrast, viral approaches tend to insert the gene within the genome, and so this makes it a lot more efficient. So I'm wondering, a tumor cell might very quickly be able to, for instance, resist this kind of approaches by basically removing those genes. Is it something fair to say, or is it you don't think it's an issue?
I just want to correct myself or to clear the confusion. All one needs is to deliver these material into the cytoplasm. That's sufficient enough to carry out the rest of the processes.
I see. So, okay.
Yeah.
That is true. At the same time, I think Sylvain brought an important point that the viral vectors, some viral vectors basically try to incorporate into the genome, and that sequence is present. Now, it's good to have that kind of a thing within the tumor cell. So as the tumor cell divides, basically the gene is switched off all the time. But the problem with the nanoparticle is that it's more transient. That means that particular population is going to get this switch off. But the question is if the cell survives after the nanoparticle products enter into the cytoplasm, then they basically lose the nanoparticle effect. But the thing is, what happens, I think the retention of this nanoparticle is a little longer into the tumor microenvironment. So that helps an uptake into the new cells as they divide. So there's a new uptake that happens. That's one of the reasons I think we might have to give more than one dosing in order to keep them keep the effect as we go on. With a viral particle, you just give one injection and then you wait and see what happens. But in this case, particularly those viral particles where it doesn't get incorporated, people give everyday injections in a preclinical experimental setting. But in this case, I think you can do the same thing, but it is much safer that it doesn't get incorporated into the genome. But at the same time, it is more targeted only to the tumor cells, not to the normal cells. And also it helps to delineate the tumor where it is present, then it can be targeted with radiation.
Rao, do you want to add a bit more on the topic?
Yes, what Mansoor says is currently being utilized, the viral approach. The one advantage over viral in the case of delivering through nanoparticle is you're actually knocking off that the cell which is supposed to be not behaving to either a radiation therapy or a chemotherapy. So to enhance some of these aspects, you deliver, like in the case of Mansoor and Alan's project, where we deliver a MDM2 component to silence the MDM2 component. So once that particular set of tissue is knocked off, I think you clear off the nanoparticle and you have less toxicity anymore. And regarding the utilization of oncolytic virus, etc., it's a totally different ball game.
Right, it is. And basically, it's not. There's a lot of issues with this approach too in terms of risk. So the question I think I would like to bring up is in terms of radiosensitization approaches in the world for radiotherapy. It seems that most radiation oncology departments are not really using any type of approaches in a regular manner to sensitize the tumor as you get exposed to radiation. It seems that there's been a roadblock somewhere in terms of putting this in practice.
Yeah, the thing is, Sylvain, one of the biggest concerns when we are talking about adjuvant radiation therapy, particularly with the non-radiation sensitizers, then that's fine. But there are sort of new radiosensitizers that come into play. So most of the medical oncologists and radiation oncologists are not aware of what is really happening at the cell cycle level with these agents. So one of the things what you do is that they just combine, like you give the chemo or an adjuvant agent 3 hours after the patient is given radiation treatment. But you don't know actually what happens in terms of because remember, the radiation responsive phase is the G2M, it's the most radiosensitive phase. But if you are, overturning the cell cycle phases more towards the S phase, then you're not going to get any killing effect. There's going to be more resistance. So having said that, if you are going to deliver this kind of a agent with a nanoparticle where you're flooding the system, which is already there in the tumor microenvironment, and then you're giving radiation, so you're really not taking out the agent away from the tumor microenvironment. It is present all through the time, and you may have a better way of targeting or overcoming the cell cycle mediated effects. So that's one possibility. Now again, we still are learning a lot of things in terms of how. There are some nanoparticles that can be basically activated to enhance the release of these agents with radiation. Now, I'm not sure whether Rao is working on that part, but there are some agents which I learned in the literature that which we wrote this in the article also, that there are radiation-activated nanoparticles that can help to either release or activate or create some kind of a heat. There's a lot of things that can happen in order to, sensitize. Now, again, the radiation, as coming to your point, the question is that at this time of the day or this time, basically you have most of the people in the field, anything that comes out from the medical oncology field, they try to combine with radiation and see the effect. Most of the clinical trials with the newer agents have not come out with any promising clinical data. It's just because there's no robust preclinical screening of those agents before it goes into the clinical trial. And that's the most important caveat now. But again, this is a completely different ballgame to talk about compared to what we wrote in the review. Here the focus is more into identification of the tumor region and demarcating the normal tissues, at the same time loading up with a therapeutic agent that can help to enhance the effects of radiation. And again, this radiation field which will be more targeted only to the tumor area, not to the normal tissue area.
Rao, do you want to add something?
Yes, we initiated when Mansoor and the other group were still in Miami, some of the possibilities of bringing in novel approaches where you actually trigger release of these components wherever this sensitization has to take place. So it's a great area to bring in multiple expertise from biophysicists and radiation expertise to come up with new venues where one can actually activate compounds to actually enhance these radiosensitization. One particular example is the known gold nanoparticle, which enhances radio sensitization. One can target the nanoparticle to the site of interest. So people in Miami, now we are in this aspect of studies to identify novel radio sensitizers.
Yeah. Okay. So, in terms of the future, what do you think would be very helpful to make this technology and those multidisciplinary team move forward? What roadblock do you see coming up, and how do you think we could achieve the work?
To answer to your question, Sylvain, the molecular imaging component, especially in preclinical, is the importance of that to blend in along with this nanotechnology to will be very useful in radiation therapy studies, especially for things like IGRT, image-guided radiotherapy, where one can utilize sensitive approaches where one can use a high-throughput screening of compounds in vivo per se, because that's one thing which is lacking in this field. At least Mansoor, who is the expert in this field, will probably give it a yes. The in vivo assessment of these compounds could only be built in when we have a robust technology which has molecular imaging, nanotechnology, as well as some of these focused small animal radiation doses. What do you think, Mansoor?
The problem, what I see, the limitations, I mean, you told the positive part, but the problem is the fluorescent-based approaches, particularly to bring this in the clinical arena, is still a long way to go. It is great to do this in a in vivo model, but in a clinical arena, I think particularly for the deep-seated tumors, it may be not as effective. The only thing I can think of is that you can use some kind of a probe-based, microscope-based approach to look into this kind of a live, real-time image that can be combined with robotic surgery to identify the normal and the. But again, you have an invasive technique being done in order to do this. So there are limitations, particularly when we're talking about near infrared fluorescence imaging-based approaches. I'm sure the depth can be improved over the time with more sensitive nanoparticles. But again, there is a good opportunity of mixing this approach with the traditional imaging approaches, and that can, synergize this field and bring out a revolutionary change in the area, the way we manage the solid tumors.
So that's pretty exciting, and maybe we can finish on this. I was going to ask you two questions. One is there any clinical trial going on right now with humans with nanoparticles, and is there any new NCI initiative for starting new big projects on this kind of approach? Maybe some special funding or?
Yeah, there is a image-guided clinical trial, RFA, that is a open. I think it's not an RFA, it's a PA, that's an open PA that is still available, where you can use the image-guided approaches to target your treatment. So I've seen a couple of grants in Miami also. I think they used MRI-based image-guided approach to look at the DC areas. And DC areas can be, targeted with high-dose radiation therapy. But again, this is not a nano-based approach. I'm sure there is a nano-based approach. RFA is there, but not in the clinical trial. I'm not sure. But there is an image-guided, but it can be through any mechanism.
Regarding the clinical, I'm not well aware of things, but some of my other collaborators in MD Anderson are testing nanoparticle-based approaches in radiation therapy. And actually, Mansoor is also aware of people.
Well, I think it's a very promising new field of research that you guys have been pushing by at least reviewing the topic. So I wanted to thank you for doing so. It was a very interesting paper. And maybe if you have any last words for the public listening to us.
I think this review article opens up a lot of ideas. Particularly, I sort of provoked the minds of different researchers in the field who is working particularly with nanoparticles, where when you want to develop nanoparticles, you might want to keep in mind that you can use it for imaging. At the same time, you can use this for targeting the tumor, where you can load a biomarker agent into it. At the same time, load the therapeutic agent into it. So I think if we all work in that area more effectively, I think not only that, like for example, in an MRI base where you have the gadolinium, can that be modified to, take up the load of therapeutic as well as, a targeted antibody. So there are a lot of things that can be looked into it. I think that we need to have more research into this field in order to, use it more in a revolutionary or a paradigm shift pattern than what we're doing currently now.
For the general public, molecular imaging is going to be one of the top 5 essential elements in the next 5 or 10 years, which might do advancement in medicine. Especially what, like Mansoor was mentioning, the portmanteau diagnostics, which is therapeutic to diagnostics, is coming into the picture, where a nanoparticle-based approach together with molecular imaging will be very useful both in discovery in a preclinical setup and translate that element into the clinics. It may be a single modality or a multiple modality. So optical, PET, SPECT, CT, MR is going to be next advancement, which whatever we right now know, we just scraped the surface of it, and there's a lot to go with this kind of marriage between biomaterials and nanoparticles. Thank you.
Well, on those last words, I wanted to thank both of you to join us today to discuss this fascinating topic, and I wish both of you all the best for your future work on nanoparticles and radiotherapy.
Thank you so much, Dad. Thank you for the opportunity. I appreciate. Thank Radiation Research for selecting our article and give us an opportunity to share our findings. Thank you so much.
Thank you, Sylvain, and thank you, Radiation Research Journal people.
Thank you very much.
Thank you.
Okay, bye-bye.